Sir, you can start, sir.
Yes, ma'am. Thank you. Thank you so much, ma'am. Hi, students. Good evening, all of you.
Good evening.
Already, what are the? Oh, yes. Good evening. So already you know a little about what are the subheadings. Already we discussed. Will you please summarize all those things? Parts of the flower. First one, isn't it? Then reproductive parts, particularly anther, and then structure of anthers and microsporogenesis, types of pollen grains, particularly what are the types of tetrad stages, how many types of tetrad stages also there, and special species of microsporogenesis, and what is meant by pollinia, and what are the example has given in the book. That things also already we discussed. And structure of pollen grains. Okay, that things. And after that, what we are going to discuss? Anybody? Any guessing? So what is meant by hibiscus bodies? Okay, that things already we discussed, isn't it? Today, what we are going to discuss, you know, see that the name of the subheading is structure of pollen grain. Already we discussed, I think. So it is also called as, what is that? Male gametes, isn't it? Pollen grain is nothing but the male gametes. What is meant by translator apparatus? Where it's present in it? And what about their functions? All those things already we discussed. So what are the types of pollen grains? So longest pollen grains in Justira. The 8 nucleated embryostic type of pollen grains is also found in Hyacinthus. This was first introduced by the name of the scientist is called as Nemec. And also viability. These things also already we discuss. And what are the elements essential for the what is that germination of pollen grains? Already that things is also already yesterday we discussed, isn't it? So particularly when the pollen grains reach the stigma, these are the content is essential. These are the nutrition substances also essential for the growth of the what is that pollen grains, particularly water. Okay, water, sugar, lipids, and protein, phenols, flavonols, B, and also calcium ions also very, very essential. Boron already told you. Boron is very, very important for the germination of the pollen grains, maturation, otherwise called as maturations. Okay, and calcium is also very, very important for the permeability of the promotes vigorous growth of the pollen teams. Suppose it is also very essential one. And then pollen teams types of pollens also depends upon the what is the plants. There are so many hydrolytic enzyme. Okay, also secreted by the tissues in stigma and style. Okay, these are the things already we discussed. It's also comes under the heading is hemotropic attractions. It is called hemotropic attraction. You know about the movement of the plant, thigmotropism. Okay, that things the hemotropism already studied. Okay, otherwise we will study in later. Okay, so hemotropism, why it is called as hemotropism? You know about the male gamete move towards the female. A female gametes also made up of chemical substances with the help of the chemicals. So that's why it is called as hemotropic attractions. Okay, this was induced by the some of the components, calcium, vitamin B, and sugar, and also inositol complexes. These things also very, very important. Already I told you, this is most of the cases. Okay, so many questions they were asked. the pollen grains can be stored like a semen of human beings for years by cryopreservation. The science, the deal study of the temperature related, what is that? Concept, that is called as cryopreservation. Particularly in liquid nitrogens at minus 190 degrees Celsius, we can store, which one a male gamete as well as a ovum or pollen grains or whatever it may be, and we can store for many years for the further uses. Okay, so these all are very, very important for the crab breeding programs. Suppose if you want to produce a new variety, otherwise called as hybridization process or seed bank, and also pollen bank also, yeah, these type of technology and they were used. Okay. Structure of pollen grains already we studied. Okay, exine, intine, and what are the content present in it, how it will be. The outermost layer is called a sporopollen. It will not easily, what is that? Degradable. Okay, that is non-biodegradable. Bio means living organism, Okay, and also not only that, and also high temperature and strong acid and alkali. It will not easily destroy. This is a structure already we saw it in the last class. Okay. So, and next, no enzymes also is there. Okay, any enzyme it cannot be easily destroy the outermost wall. So that is also a very, very important. And then another one is, this is also sometimes they may ask in the match the following questions. Largest pollen grains, mirabilis, already yesterday we discussed. Smallest pollen grains, myocytes. Think both are in mm. At the same time, myocytes is the smallest one. Longest pollen grain, Justira, the micrometer will be 2500 to 3000 micrometer, and thread-like structure, as well as called as a ribbon-like structure is that. I will show you. And palynology, what is the meaning of palynology? It is also another thing. What is my pomology? What is my pomology? Anybody else? Anybody else? What is my pomology? The science that deals with the study of fruit is called as, what is that? Pomology. Okay, here pollen grains means palynology, not paleontology. Okay, paleontology means the science that deals study of fossils. Okay, here the geopaleontology, see that fossils, pollen, geopaleontology. It is also nothing but the pollen study of the fossils, pollens. Aeropollenology, it's found, pollen grains found in air, because aerosols, there are so many. What is that? Pollen grains, it may be easily spread through the air. As a result, what happened? There are so many allergies also happen to human beings. Already we discussed that things also, isn't it? Okay. And also another one is called as a medical aspects of the pollens. Okay, this one is also we here as given. Okay, see that patropaleontology. Melitropaleontology is the study of the pollen found in the honey. Okay, because all the honeybees collect the nectar. Okay, honey collected from the nectar. So along with the which one pollen, isn't it? That's why it is also another one. See, this is the structure of Mirabilis jalaba. Here the what is that? The pollen grains is a very long nature. Sorted form, Mirabilis jalaba. This is a sorted one. This is also myocytes. Sorted pollen grains. Here, very small one is there. Here is the longest one. Okay. So this is also another type, heterostyle. What is by style? Where it present already, but it's mostly present the, what is that? Female gametes. The upper part is called as, what is that, style, stigma, and ovary, like that, But here, anther, it has a long style. Some of them, it has a short style flowers. These are the two examples has given. This is the longest one. This one is the shortest one. Okay, that's why it is called as heterostyle style. Okay, so this is also sometimes they were asked. And then very, very important things already, about each and every pollen grain, there are some germ spores also present. The germ spores exist in three forms. That is, one is called as lens and also slit type and also circlite type are present in the pollen grains of the dicot, particularly capsular. For example, dicot plant, these are the kinds. Okay, these are the kinds of germ spore also present in it. Example, capsula. Suppose if it is present in these three type of the germ spore in a single plant or same plant, this type of pollen grains are called as trichoplate. This is called as trichoplate. Okay, because here lens shape, slits type, and also circular type. They have given in the structure in the lower diagrammatical representations. See, the first one is called a circular germ spore. They see this is the pore. Okay, just a small opening is there, but here the structure will be in circular manner. That's why they have given as a name is called a circular germ spore. Second one is called as just like a lens in the biconvex or concave. This one is called as biconcave like structures, lens. That is Colpus germ spores. It's also called as Colpus germ spore. Okay, and then third one is called a germinal furrow, just like a groove. Groove means it's nothing but a small. What is that? What is a groove? Is nothing but a what is that? Down. Okay, so this is called as germinal furrow. So the germinal furrow mostly found in monocot plant, but these two are mostly found in the dicot plant. That is called as sporate pollen grains and colpate pollen grains. These are the two types. These are the two types of germ spores mostly found in dicot. This is also so many times they were asked in the questions. Okay, so this is another things. And one germ spore is present. Suppose if it is one is present, that is called as monocoplate. It's mostly present in monocot. One monocot and monocoplate. In other things already you know about. The plant is in which pollination takes place by insects. The pollen grains having only one layer around the pollen grain, it is called as pollen kit. Suppose if it is present the only one, what is that? Layer around the pollen grain, that pollen grain collectively called as pollen kits. It is composed of lipids and carotenoid. This is very, very important. Pollen kits are made up of which component? It is composed of what are the components or biomolecules? They may ask the questions. Okay, and also examples. Okay, lipids and carotenoids. Already you know about carotenoid. Already we discussed it. It is nothing but the vitamin A, isn't it? Okay, lipid, because it's also one of your fat soluble vitamins. Okay, but here lipid and carotenoid. That's why it is how it will be. The pollen kits, it is not easily react with the water molecule. So it is a sticky nature, isn't it? Just go and touch any one of the flowers. Okay, you touch the pollen grains, just like a jelly-like structure is there. Okay, it can be easily semi-solid. Okay, that is called as what is that? Lipid and carotenoids. Okay. Say functions of pollen. What is the main function? Suppose the presence of lipid as well as the carotenoid. What happen? What is the use of pollen grains? You know, see, this oily protect them pollen grains from the harmful ultraviolet radiations. This is also very, very important. Suppose say sometimes they may ask the question. The oily protect them pollen grains from the harmful ultraviolet. Instead of ultraviolet radiation, they may ask the questions infrared radiations. They may ask the questions. So be careful this area. Okay. And sticky surface help to attach with the insects, because suppose the surface is sticky in nature, easily what happen when in the insect come and sit on it, what will happen? It will be easily attached, because the about already we discussed Asclepiaceae, the polonium also easily carry out through their legs only. Okay, so it is also easily attached with the insect. It's a yellow in color, attract the insects easily, because it is a yellow in color. Pollen kits always in yellow in color because the presence of carotenoid. So it can easily attract the yellow color. Okay, easily attract to the insect easily. And then the pollen kit is present in the pollen sac. Always the pollen kits mostly present in the pollen sac of capsula. Here the angiosperm gas given in the examination aspects. Sorry, example aspect. Sorry. Then we'll pass on to the next one is called dehiscence of anther. Dehiscence means how it will become. It is essential for the pollination process. Suppose it does not happen. It is not dehiscence of the anther. What will happen? The pollen kits or pollen grains. It will not come out. It will not reach the stigma. As a result, there is no pollination is possible. So what will happen inside? How it will be dehiscence takes place? So that things we are going to discuss. After the maturation or during maturation of the anther, various changes takes place in the wall of the anther. Already you know about. Okay, what are the things? Middle layer completely. What is that? Degenerated. That things came next line, isn't it? In the beginning, the middle layer of degenerated due to the absorption of food by tapetum. Already you know about the things. So, what are the layer present in the anther or dehiscent anther means epidermis, endothecium only present in 2 layer only present in it. See here, next line is that. In a mature anther, only the 2 layer present, epidermis and endothecium, present in the form of outer covering. These 2 layer only present in the mature anther. Mostly they are asked the question like this. The sterile tissue present between the both the pollen sac of the each anther lobes degenerate. Suppose if it is present in the, what is that? Pollen sac in between, there is a sterile tissues present in it. The sterile tissue is also completely degenerated. So both pollen sac of each anther lobe fuse together to form a single pollen sac. So in between there is a sterile tissue. It is completely degenerated. As a result, what happened? The two, what is that? Anthers lobes fuse together, form a single pollen sac. So each and every mature anthers, only 2 pollen sacs are present. They may ask the question, the mature anther containing how many pollen sacs are? Only 2 pollen sacs are present. That is also very, very important. Okay, so this is the structure. So this is the outermost layer is called as, what is that? Epidermis. What happened in it absorb, what is that? Which one you know? The middle layer. See that? This is a middle layer. It absorbs the nutrients. So as a result, what happened? It will be easily destroyed in this area only. So these are the, what is that? Sporogenous tissues. Inside there is a tetrad stages there. This is also a connective tissues. They already we studied that thing, so isn't it? Okay. See that? This is called as pollen grains. Inside there is a pollen grains. The outermost there is a cover is there, that is called as pollen sacs. This is another structures. Okay, already see that things. So this is the way. See that inside, this is the, what is that? Anther. When you are going to take the cross section, you can see these type of structures. Inside what happened? Okay, these are the outer layer, connective tissues, epidermis, endothecium, sorry, middle layer, tapetum, and stomium. Okay, this area only, already I told you, where is the dehiscence takes place? Mostly the stomium region, this area only. Then what happened? The mature pollen grains, it comes out and reaches the stigma. Okay, when you are going to see under the microscope, the structure will be like this. It is a groove-like structure. There is a groove. Okay, it's made by the absorption of water molecules and from the, which one you know, a middle layer by or tapetum. Nutrition also absorbed by the tapetum. As a result, it made it make it as a groove-like structure. As a result, it may be split. Or what is that? Dehiscent. So what is the main mechanism of dehiscence here? How it will be happen? Okay, when dehiscence of anther takes place during the dry season due to the hygroscopic nature of endothecium. Hygroscopic means water lossing, isn't it? Okay, loss of water takes place from the cell of endothecium. So what happened? The outermost layer completely dry in nature. So as a result, it make a curve-like structures. Okay, this is the first step. And wall of endothelial cell try to contract due to the loss of water, but inner and radial wall do not contract due to the presence of future thin-walled cells of fibrous thickening, whereas outer thin-wall cells of endothecium contract and become a concave or incurved. That's why I told you The innermost layer present as it is, but at the same time, the outermost wall is contract. As a result, shrink. You can tell as them shrink. Okay, due to the loss of water molecules. Finally, what happened? It make a incurved natures, otherwise called as concave. Okay, it's just like a bending. Okay, what are the types of dehiscence in anther? Different types of dehiscence also takes place here already. So the diagrammatical manner. So these three, see, this is called as intros. Okay, so longitudinal. This is also longitudinal. This is also longitudinal. These three are longitudinal aspects that dehiscence takes place. Okay, see that the center portion there is and the dehiscence takes place. This is also laterals. Laterals means lateral side. It takes a longitudinal aspect. And the extros longitudinal. Extros longitudinal in this area. In this area. Okay. See that? Yeah, longitudinal aspect. Transverse means in between, middle of the anther. It takes place in transversely. Porous means the top of the anther. The head of the anther like that. It is a head like, okay. So here, what happened? There is a small pore-like structures. It may be dehiscence like that. This is a special type of dehiscence takes place in the anthers. This is called as valvular. Valvular means, see that the structure, the outermost layer completely, just like a what is that? Opening of the outer layer. Okay, that is called as valvular. These are the dehiscence of anther types or kinds or whatever it may be. You can call that. See that another same things is this is called as transverse. It is also a transfer. The pollen grains or comes out. This is longitudinal and poricidal. See that poricidal. Poricidal means nothing but a small spore on the top. The microspores are comes out. Okay, valvular. Already I told you, just like a opening of and closing of the thing. Your eyelid also like that. See. In curving of outer walls, after that, in curves also made as a result, what happened? In curving of outer walls, exert pulling force or tension over the entire surface of the anther. Okay, suppose it's made a curves means it give a what is a surface tension. The outermost wall layer completely what happen? It may be shrink. Due to the tension, thin wall stomal cells break off. That is very well. Which cells are break off? Stomal cells are breaks off, and dehiscence of anther takes place, and pollen grains are present in pollen sacs released into the atmosphere. Okay, so dehiscence of anther is the most of the angiosperm is longitudinal. This is very, very important. Take an example. Most of the angiosperm, how it will be dehiscent. The longitudinal aspects. Dehiscence of anther of capsula is also a longitudinal. That's why they have chosen for the, what is that? Typical angiosperm. Got it? I think that's all about the first part. Okay. So we'll pass on to the next one. Okay. We will pass on to the next one. Okay. See the part 2 we are going to discuss. Okay, see that part 2. See. Here, what we are going to discuss, already what are the things already we discussed? First of all, flower and male part of the flowers. What are the microsporogenesis, microsporangium? That things already we discussed. Next, what we are going to discuss? See that. Say microgametogenesis. Gametogenesis means gamete inside the pollen sac mother cell. Male gametophyte is also produced. Microspores also produced. This is also already discussed. In flowering plants of the flowers, pollen grains or microspore is considered as the first cell of male gametophyte. Pollen grain comes out, that is the first cell of male gametophyte. When it reaches a stigma, before the what is that? Events occurs, that things we are studying. Partial germination or development of the pollen grain takes place before dehiscence of the anther is called as Previous development. Suppose before dehiscence, the anther is called as what is that? Previous development. Okay. Development of pollen also takes place at mother's places. Inside the pollen sac of mother is called as in situ development. Okay. This is also previous development. This is also called as a in situ. In situ means within the mother cells, mother pollen sac. Mother pollen sac. Okay. So next one is, what are the changes? Pre-pollination development. Before pollination, what are the changes occur within the pollen grains? See that in the beginning of the process, only nucleus of pollen grains divided by unequal mitotic divisions. Once the mature pollen grain is there, right? Okay. After the maturation, the pollen grains comes out. Before pollination, pre-pollination development. Okay. In between the pre-pollination, okay, what are the development changes within the, what is that? Pollen grain. First, in our lab, there is a two nucleus present inside. The two nucleus also divide. How? Unequal mitotic division resulting two unequal size of nucleus are formed. Okay. When you are going to study the structures, you can see the, what is that? Diagram, you can see that how it will be divided. I will show you later. The small nucleus present near the wall is called as generative nucleus. Note it down. This question also asked in the NEET question paper. Which nucleus present near the cell wall? Small nucleus. At the same time, it is a generative nucleus. And the large nucleus present inside the cytoplasm is called as true or vegetative nucleus. You must note it down which one is the largest nucleus in the pollen grains. Okay, you have to tell us the, or otherwise called as pollen tube. They may ask the questions. Vegetative nucleus, otherwise called as, what is that? True nucleus is called as, what is that? It is a largest one. This is the smallest one. This is the largest one. It is near the wall. It is present in the cytoplasm. Okay. Both the nuclei are surrounded by a cytoplasm and is become dense, then followed by unequal cytokinins. Cell division only. That is otherwise called as mitosis, isn't it? That's why here given as a mitotic divisions. Resulting to the cell of unequal size or form. Already knowable. So inside what happened? After the maturation of pollen grain, before the pollination process, what are the changes occurs within the cell? A single cell undergo the mitotically unequal size, large nucleus one, and also small nucleus. The large nucleus name is called as vegetative nucleus. Okay, and small one is called as generative nucleus. Okay, see. Go to the next one. The large cell, which a large nucleus is present, known as vegetative cell or tube cell. This is also called tube nucleus. And smaller cell in which is a small nucleus present called as generative cells or generative nucleus. Okay, this is already no. Now pollen grains come in bi-celled stage, isn't it? Pollen grain, how many cell stage is the bi-celled stage? Because it's a nucleus present in two. Okay, and all the mitotic division also takes place. This stage of pollen grain is called as partially developed male gametophyte or mature pollen grains. Already we discussed all the angiosperms. Which cell stage is mostly present in bi-celled stage? So mature pollen grains always present which cell? Bi-celled, of binucleated nature. Okay, see the next points. In over 60% of the angiosperm pollinations, the pollen grains takes place in bi-celled and binucleated stage only. This is very, very important. So many times they were asked. Okay, bi-celled stage. This is also very important. Otherwise called as binucleated. How many percentage they told? 60% of the angiosperm pollinations also bi-celled and binucleated stage have been reported by many scientists. There are many scientists have been reported and analyzed. Then, and in the remaining angiosperm pollination occur at 3-celled stage. This is also possible. Remaining, how many is there? 40%. In these generative cells, note it down. In these generative cells divide and form two male gametes. The male gametes also formed from which cells? Generative cells. Okay. So the male gametes, generative cells. This is also a very total. How many cells are there? No, already in our board. What is the vegetative cell? Is the one. Here the generative cells are divided into two. So total 3 cells is there. Okay. So this is also a very important things. Okay. The development of pollen, which takes place inside the sporangium, is also known as endosporic development. Suppose the pollen grains development takes place outside the sporangium is called as, what is that? Exosporic. But here always takes place inside. Already we discussed only in the, what is that? Sporangium only. Development always takes place in the sporangium inside. That's why it is called as endosporic development. Sporic development. Now, generative cell changed into vermiform of spindle-shaped structures, which cell? Generative cells. Okay, got it on main point, which dense cytoplasm and detach from that wall and enter inside the vegetative cells. These are the very important things. Okay, so go to the next one. And then post-pollination development. After that, what will happen? Further development of the pollen grain takes place on the stigma. Once it reaches the stigma, what happen? Or carpels. Okay, already told you. The stigma contain moisture, sugar, and also so many vitamin B and inositol. There are so many components. This is there. Okay, it is easy. Water molecules also present in the stigma. This is the female reproductive part. That one is called as male gamete. Male gamete, when it contact with the stigma or carpel, what happen? After pollens absorb moisture and sugar content from the stigma. Due to this, the volume of cytoplasm increase. This is also so many times they were asked. Okay, when the pollen grain reaches the stigma contain these are nutritive substances and also water molecules is also there. It is absorbed by the, which one you know? Pollens. As a result, the inside the cytoplasmic volume will be increased. Okay, it exert pressure. That time what happened? Automatically it exert the pressure on the both outer layer, isn't it? So this is the initial reactions for the formation of pollen tube. See, because of this pressure, intine comes out. Note it down, very important. Not the exine, because inner layer is called as intine, isn't it? The intine comes out through any one pollination. Germ pores in the form of tube-like structure is called as pollen cake. This is very important. Okay, the pollen tubes also are originated from which layer? Means intines. Okay, which excuse me. See. So this is the structure of pollen grains. Binucleated stage. This is called as, what is that? Generative nucleus. This one is called as tube nucleus, isn't it? So this is a pollen grains. The outer layer is called as exine. This one is called as intine. When it reaches the stigma, what happens? It absorbs the nutrition substances. As a result, see that this is here called as exine form in the form here gives rise to pollen tubes. Okay, this is the way the nucleus so divides. Generative nucleus, here is just take it an example. This is the generative nucleus. Here is the one is there, here is also divided, isn't it? The tube nucleus, this one is the largest one. It's moved towards the, which one you know? Ovule or ovary. Okay, this is the basic structures. See, this is the way. See here, this is a structure. Germ pore is that. This is nothing but a space like the structure is the germ pore. It's germ. Okay, the pollen sac. It contains the small pore-like structures. Okay, and also the inside what happened? You know, there is a layer exine. This one, sorry, exine is outer layer. Intine is the inner layer, and nucleus is also present inside. This is also called as cytoplasm. Next one, see that what happened here. See, generative cells division is also takes place here. Just go to these are all are same, but in this category, what happened? You know, the intine form as a tube-like structure. This is called as intine. The inner layer is called as a intine. So these are the two nucleus comes out. So finally, what happened? The pollen tube formation takes place. The pollen tube formation takes place. Here, what happened? The single nucleus, which nucleus? Generative nucleus divided into male what is that? Cells, otherwise called as male gamete. We may tell as the male gamete. This one is called as what is that? Tube nucleus. Tube nucleus. What is that? It is give you a guidance for these two things. Okay, these two male cells. See, first of all, vegetative nucleus enter into the pollen tubes and assume terminal positions. The spindle-shaped generative cells now enter into the pollen tubes. First, before formation of the pollen tubes, first which one is enter? Generative cells only enters. Inside the pollen tubes, generative cell divide mitotically and to form a two non-motile male gamete. Now, male gametophyte come in three-celled structure in which one vegetative cell, sorry, here the spelling mistake is there, and two male gametes are present. This is very, very important. Which one? Male gametophyte, one vegetative, two male gametes present. Okay. Go to the next one. This three-celled stage represent the mature male gametophyte. Note it down. This is a very, very important. The mature male gametophyte, how many, what is that? Cells are present? One vegetative and two, what is that? Generative cells. The two generative cells is called as male gametes. Okay. The male gametophyte is highly reduced and completely depends on sporophyte. This is also another process. Sporophyte is nothing but the plant body. Okay. We'll see. For the formation, these 2 points is very, very important. Note it down so many times, 100 and 1000 times you can make it as a mock. For the formation of mature pollen grains, one meiotic and one mitotic division is required. For the formation of mature pollen grain, one meiotic, one mitotic division is required. At the same time, for the formation of mature male gametophyte, one meiotic and two mitotic divisions are required. Here 1:1, here 1:2 ratio is essential. Where here the male gametophyte, here the pollen, mature pollen grain. This one is also very, very important. Okay, see. So these are the structures. Already you saw it. Okay, this one is called as a megaspora. Megaspora is nothing but the female reproductive parts. Otherwise called as what is that? Megasporogenesis. Otherwise called as gynecium. Okay, this is the structure of pollen grain and pollen tube formation. How it will be reached? This one is male, this one is female. Here the 2 nucleus is there, here the 8 nucleus is there. How many cells are there? 7 cells are there. We are going to study one by one, isn't it? Okay. Okay. Up to this level, you have any doubt? Do you have any doubt in this area? Students? No. Okay. So let's pass on to the next one is called as female reproductive organ. Which one is the female reproductive organs? Already you know about gynecium. Gynecium is the female reproductive organ, isn't it? Okay. The free unit of gynecium is called the pistil or carpel. This is also very, very important. Carpels. Suppose we are going to study, okay, the fruits, structure of fruits. Okay, that time the monocarpellary, bicarpellary, polycarpellary. Okay, so it's also depends upon their gyneciums. Carpel is also known as megasporophyll because the size will be very large in it. That's why it's as hence it is called as what is that? Megasporophylls. Okay, the carpel contain how many things? There are 3 distinction region is there. One is called a stigma and style, and third one is called as ovary. These are the 3 things mostly present in the megasporophyle. The free end of the carpel which received pollen grains, this is called as a very, very important. Pollen grain is called as stigma. Okay, see the diagram. This is stigma. This is the receiving point from the male nucleus. Just here you have to draw the which one you know. That what is that? Anther. Anther, how it will be developed? and the digestion takes place, and also it's also what is that transfer from anther to stigma. It's a female, isn't it? The uppermost layer is called as uppermost part is called as what is that stigma. The middle portion style already in our about. This is called as ovary. Okay, collectively this inner layer is called as ovule. Okay, and this is a inner layer. This is called as nucellus. Inside, see the integument is there. There are two integuments, inner integument and outer integuments. Okay, so inside how many cells are present in it? 8 cells. Okay, 8 nucleus is there, but 7 cells and 8 nucleus is present in it. We will see the detail. This is the normally female. See the structures. Okay, so these are the structures already saw it, isn't it? Filament anthers. The anthers releases the pollen grains. It reaches the stigma, and pollen tube formation takes place and reaches the where? In the ovary or ovule by the process called as pollen tube. See that pollen tube is also given here like this. When it reaches, what happened inside? That things also we will study. This is a receptacle. Receptacle is nothing but a small stalk-like structures. Okay, it is attached with the which one you know? Ovary. See that a long narrow tubular structure is present in the between the stigma and the ovary called as tail. Already in our report. The basal swollen part of the carpel is called as ovary. That is already in our report. The ovules are also known as integumented. Why? Because there are 2 or 3 layer covered by a megaspore. That is called as integumented megasporangia, which are born on the cushion-like tissue called placenta. This is also very, very important. It's a lower part. There is a bed-like structure, cushion-like structure present in it. On that, there are all the parts is present. There is placenta, that is called as thalamus. Okay, in the ovarian cavity. Ovarian cavity. These are the very, very important. What are the examples has given, you know, here? 1, wheat and paddy and mango. Or more than 1, papaya, watermelon, orchids. Ovules are present inside the ovary. How many ovules are present? 1 ovule present in wheat, paddy, mango is all having only 1. Take an example, papaya, watermelon, and orchid. These are the fruits, isn't it? These are the, what is that? Mango is also one of the fruits, but particularly these are all considered in and cereals. Okay. So ovules are present inside the over here, more than 1 present. See that? So this is called as, according to the carpels, according to the nature of the carpels, they may be divided into 2 categories. One is called as apocarpus gynoecium. You take it an example, a free carpels. See the first diagram. See, these are the, what is that? Carpels. These are all free, isn't it? It is not united with the any tissues or together, isn't it? This is called as apocarpus. Next one, see here, this is also united. Take an example, so cucumber. Okay, here given as here, what are the apocarpus gynoecium free carpels? Rose, lotus, and michelia. These are the examples of apocarpus. That means carpels are free. Okay, syn means united. Syncarpus gynoecium. Syngame, gametes are united. Okay, fused carpels, papaver and hibiscus. Papaver somnifera. Papaver means nothing but the, what is that? It is not a pepper. Okay, papaver somnifera. So many plants is also having the same structures. These are the diagrammatic representations. Okay. Here the same things is there, but here the tetracarpal nature. This one is, each one is considered a carpel. 1, 2, 3. This is called as tricarpal. Tricarpals. Okay, this is also tricarpal, but is united. This one is free nature. What about here? Here tetracarpal. See that? This is the tetralocular, otherwise called as tetralocular. This is also called as a trilocular. This is also a unilocular. Here, unilocular. All are united together without any cross-wall formation. There is no any connective tissues here. Here, there is a separate. Okay. So see the, this is the female, just mostly present the hibiscus. This is the uppermost stigma is a, what is that? Pentamerous nature. It's also divided into 5. 1, 2, 3, 4, and 5. This is the long tail is there. All the tails are united in the bundle of sheath like that. There are so many, what is that? Anther present in it. finally there is a ovary. The bottom of the ovary is called as thalamus. Okay. Here the stigma also given here. See that second one, stigma like this. It is a united one. Otherwise called as multicorpulary. This is called as multicorpulary. So this one is also another one. Okay. This one is also called as the multicorpulary apocorpus gynecium. Micellia. This is an example of micellia. Okay. The last one is called as the structure of ovule. What are the parts present in it? We will see the next slide. Okay, the placenta is located inside the ovarian cavity, arising from the placenta or the megasporangia, commonly called ovules. The number of ovules is an ovary may be 1 wheat, paddy, mango, to many papaya, watermelon, and that's already we discussed that also. Okay, see, megasporangium, otherwise called as structure of ovule. This is very, very important. So many times they were asked, what are the types of megasporangium? How it will be classified? What are the basic principle is that? Like that they may ask. Each ovule attached to the placenta by means of a thin stalk called funicle. I will show you which is called as funicle. I will call you, otherwise called as funiculus or funiculum. Okay, ovules attached to the placenta by means of stalk called funicle. Okay. The point of the attachment of the funicle with the ovule is called as hilum. There is a point. The point is attached with the funicle. Okay, the placenta attached with the stalk funicle. The funicle attached with the point is called as hilum. The main region of the ovule is composed by mass of parenchymatous cell is called as nucellus. Outermost layer. This is covered by a, what is that? Ovule composed by mass of parenchymatous cells. It's also very, very important. That is called as nucellus. The nucellus is the main part of the ovule. That is very, very important. It is covered inside. What are the things present in it? Nucellus is very, very important. The nucellus is covered by 1 or 2 coat called integument. This is also further divided into what is that? Outer integument, inner integument, like that. Okay. See the diagram. You can easily see the structures. See, the placenta is there. Here, the placenta. Where it attached with the stalk-like structure is called as funicle. Okay, the funicle attached with this part is called as hilum. Okay, okay. See that this is called as a outer integument. This one is called as inter integument. Okay, so inside there is a, what is that? This is called as a nucellus. This outermost covering is called as nucellus. Okay, there is a space is there. This is called as microfile. Microfile as well as hilum is also very, very, very important. So many times they were asked fruits, seeds, and ovule. Okay, the microfile as well as hilum. These three mostly found in these three. What is that? Things. See, next one. If a ovule of the most of the plant is attached to the main body of the ovule for some distance to form a ridge-like structure known as rape. This is also another ridge-like structure. I will show you how it will be. And vascular tissues are present inside the funiculus, which supply from the placenta to the body of the ovule. Base of the ovule is called as chalaza. The lower part is called as chalaza. Integument is absent just opposite to the chalaza. So that is narrow passage or pore is formed, which is called as microfile. Microfile is nothing but a small pore-like structures. How they classified? How they are classified? Ovules. Okay, how many types of ovule present in it? How they are classified? You know, the basic things is these are the things: microfile, chalaza, and also integument. These are the things. See that next one. Microfile is bitechnic. What does it make? Tegment. Suppose it is present, the two tegment is called as what is a bitechnic. Ovule has two-part outer region with this surrounding by outer integument is called as exotome. The outer integument name is called as exotome. Endostome means inner side. The inner integument is called as endostomes. Okay. In the most of the angiosperm, entire part of the nucellus is utilized by developing embryo sac. Okay, nucellus utilized by the developing of embryo sac. But in some of the angiosperm, some part of the nucellus remaining inside the ovules. That part of the nucellus present inside the seed in the form of a thin layer known as perisperm. Perisperm is nothing but what is that? It is a what is that? It is nothing but the embryo sac. What is that? Remaining part of the embryo sac, otherwise called as remaining part of the new cell is develop into perisperm. This is also a important one. Okay, sometimes they may ask the question: perisperm is the development of which one? New cell and also exostome or ovary, like that they may ask. You have to tell us the new cell is. Okay, remaining new cell is inside present in it. In particular, in some of the angiosperm, they may be called as a perisperm. Okay. Okay. The perisperm is commonly found in Piperaceae, Piper nigrum, and Gingibraceae families, turmeric and ginger and beetroot and castor. These are the very, very important examples. Please note it down. Perisperm, otherwise you know very well. Extra present nucellus form as a layer-like structure is called as perisperm. It's mostly present in the angiosperm. The perisperm commonly found in what are the families? What are the examples? Means Piperaceae, Gingibraceae, turmeric, ginger, and also beet and castors. These are the examples. And another one is some filaments are attached with the funicles. Sometimes placenta are known as obturatus. This is also another one. Some filament are attached. Filament is just a thread-like structures present in the placenta and funicle. With the funicle, okay. So that is called as obturatus. This is also a law, I think, asked in the 2020-21. Just go and refer the lesson question paper. You can get the answers. The functions of the arperator is the guide the passage of pollen tube towards the microfile inside the ovary. What is the main functions of obturatus? See there, it guide the what is the pollen tubes towards the microfile inside the ovary. This is very, 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 very important. Okay, see, these are the entire structure of the anatomy of the ovules. See here, this is called as microfiles. This way only the pollen tubes they can enter easily. Another category is also there. Not only in this way. Okay, so many ways is that. That's salsogamy. This area is called as salsa. The opposite of the microfile is called as salsa. So see the salsa, there is a what is that? New cellus is also present. You see that one. This is called as funicle. This one is called as xylem. Okay, this one is called as xylem. Vascular stand. This structure is called as vascular stand up. See that? This is called as raphe. This is called as a raphe. Okay. This is the outer integument and inner integument. Inside there is a 8 cell is there. Okay, the 8 cell with the 8 nucleus is there. This is also male gametophyte or female gametophyte. This one is called as female gametophyte. Okay, otherwise called as ovary. 8. The center there is a synergetic cell is the center. What is that? Sorry. Synergetic cells or egg cells. This one is called as synergetic. Egg cell present in it 2. That is the yellow color is the egg cell. Egg cell is 2. Synergetic cell is also present in it. This is called as what is that? Polar nucleus. This is called as polar nucleus. The polar nucleus is also very, very important. The polar nucleus is also here. 2 is that. The center it's occupied. The antipodal cells also it consists of 3 nucleus. 1, 2, 3. 3 cell and 3 nucleus. Okay. So total 3, 2 and 2 total. How many cells? How many cells are there here with the synergetic cells also? That's total 8 cells. 1, 2, 3, and center there is a 2, and then antipodal is also here 3. It collectively called as what is that? What is that? Anatomy of the ovule. This is called as embryo sac. This outermost layer is called as embryo sac. Okay. So see that these are the structures. This is very, very important. Just to go through it, you'll get the ideas. Okay. With that, listen. See, already we saw it. This is xylem. Okay. And funicle, microfiles, microfilar poles, outer integument, inner integument, nucellus, the embryo sac, and salsa. Sometimes it may be inverted manner. Okay. See, according to the ovules, according to the type of ovules, they may be divided into 6 types. This is also very, very important. They ask the questions on the basis of some mutual relations between funicles, chalaza, and microvilli. This is also so many times they were asked. Excuse me. Okay. The types of ovules also based on their which principles, which components lies about the funicle, chalaza, and microvilli. How many types is there? There are 6 types is there. One is called as atropous. Otherwise called as orthotropous. Second one is called as anatropous, campylotropous, hemianatropous, amphitropous, circinotropous. These are the 6 based on the which one funicle, chalaza, and microvilli arrangement. These are the 3 part is very, very important. According to these 3 only, they may be exist in 3. What is that? 6 types. 6 types of ovules. See. So how it will be? The types of ovules maximum. The orthotropous, it is a straight in nature. That means chalaza, funicle, and also what is that? And microvilli. These are all present the straight manner. Anatropous, that is called as inverted one. It may be small bend-like structure. Hemitropous, hemianatropous. Anatropous structure is also hemi means in partial manner. And campylotropous, it is a curved one. Amphitropous, transverse. Transverse means it look like a T shape. Okay. Circinotropous, these are the 6 types of ovule exist in the presence of the microvilli, funicle, and chalaza. Okay. See the diagram, you can easily understand. This one is called as ortho, straight. See that? Inverted manner. And also curved. This is called as what is that? Hemi. Amphitropous. It is not considered as a campylotropous. These are all are the structure. They ask the questions. Suppose they may give the structure, any one of these structures, they may ask the question which type, like that they may ask. You have to practice it. Okay. You must know that which one is straight, which one is inverted, hemi, anatropous, and curved, transverse, and circinotropous. You must know the position of the ovule. At the same time, examples also is known. Okay. This is also another important thing. Same things also given here. Okay. See. So these are the example. Atropous or orthotropous, maximum polygonum, otherwise called as piper, and zygus. These are the 3 examples given. PPC. And anatropous, malvaceae, compositae, cucurbitae, solanaceae, and castor. Triple C, MS. Campylotropous, cruciferae, leguminosae, and also caparidaceae. These are the 3. Hemianatropous, ranunculaceae. Okay, ranunculaceae is nothing but the citrus family. Primulaceae, this is also another one. And amphitropous, alimceae, butomaceae, centrospermiae, lemnopapae. Okay, these are the another one. Last one, circinotropous. Here, what is that? Opensia, Cactaceae, Opensia. Okay, these are the example given. Just to go through it, very, very important things. Okay, now we are studying which one Gynoecium, megasporogenesis. After the megasporogenesis, what will be the fertilizations? What are the fertilization event takes place? Okay, here according to the integument, according to the position of the ovule, they may be divided into 6 types in this category. On the basis of integument, they may be divided into 4 types. Unitech technique, that means consists of single integument. Bi means 2. Tri means 3. A-tech means absence of integument. Sometimes it may be absent. This question is also so many times absent. And tri technique also. A-technique means absence of integument is called as A-technique. Example, Loranthus and Santalum. Sandalum is nothing but the, what is that? Sandal. Okay, it is also comes under the Verbinaceae family. Verbinaceae. Okay, this also so many times they ask. Lychee. Lychee is comes under the heading on tri-technique because it contains two tegments, that is inner and outer. Here, absence of tegment. It is completely absent. Okay, here two. Here only one is there. Maximum 3 only present in it. Okay, and then so special ovular structures. These are the another things. We will finish it up to this level only. We'll see the remaining things on the next one. See that special ovular structures. Other than the ovules, some special structures is also present in it. It's also reported by some of the families. Okay. See that carnicles and also strophiole. It is formed as an outer growth and outer integument in the microphylla region. Example, Iferbaceae. Iferbaceae means nothing but this family maximum presence of which one in a latex vessels. Take an example of rubber, Hevia brasilensis. Okay, and Iferbaceae take an example of which one you know, and so many plant is there. Okay. Iferbia tricoli, Iferbia antiquorum, like that it comes. Iferbaceae family. Aril, it is regards as a third integument and arises from the base of the ovule and surrounded the ovule. Example, lychee. Already we studied that. Myristica and passiflora. These are the examples. Aril is also another one. Third integument is otherwise called as aril. Like that, you can calculate it or you can think that one. Perisperm, it is unused nucellus present on some seeds. This is also already we discussed. Perisperm, unwanted, that means extra. Okay. Black pepper, beetroot, canna, and mirabilis, and pinus mirabilis. These are the things. Okay. These things we will finish it today classes. Today, what are the things we already discussed? Megasporogenesis. Okay. Megaspores, what are the structures of ovules? What are the types of ovules? And a special type of integuments, types of integuments, and also special ovular structures already studied. Before that one, how it will be, and after that ovule structure finish, we'll enter into the fertilizations. What are the fertilizations? What are the events of fertilizations? How it will be? What is by double fusion, double fertilization, triple fusions, that things. After that, what is by pollination? Okay, pollination, double fertilization, triple fusion, and what are the changes within the, what is that, ovary after the pollination process. These are all we are going to see the next classes. Before that, if you have any doubt, please ask me now. Sorry, here in this portion I couldn't able to understand. Anybody else like that? Yes. Hello.
No, sir, no doubts from my side.
So you once again you practice it with the diagram itself, guys. Please go through the lesson and also draw the diagrammatical diagrams and all the chart and examples, all those things. Once you practice it, then it will be better. Okay. Again and again, I will tell you where is the bold letter and chart diagram question. You don't forget to practice. Okay. Shall we finish it? Okay. Thank you, my dears. Thank you so much.
Thank you, sir.
Thank you, ma'am.
Thank you, sir.
Thank you, ma'am. Thank you so much. Bye.